Hello and welcome to this lecture. Today we're going to talk about generating new glucoses from non-carbohydrate molecules or gluconeogenesis. Gluconeogenesis takes place in two important organs. What are they? They are liver, mostly in liver, and about 2 to 3% in kidney. So the primary organ for gluconeogenesis is the liver. Now let's talk about how this pathway and how we produce glucose from non-carbohydrate molecules. First and foremost, we should talk about glycolysis. Why? Because scientifically it's not correct to say gluconeogenesis is the reversible of glycolysis. It's not scientifically true, but somehow they are so similar in many pathways, because in glycolysis we use glucose and in gluconeogenesis we produce glucose. Let's start from glycolysis here and then we continue with gluconeogenesis. So this is our 6-carbon molecule glucose, right? And this is our cell. What is this beautiful cell here? For example, it can be liver. So in liver, whenever glucose wants to enter the liver cell or hepatocyte, it requires a transporter. In liver, it is GLUT2 or glucose transporter 2. Glucose transporter 2 helps glucose in order to enter the cell. Whenever this beautiful glucose enters the cell, it turns into glucose 6-phosphate, and this is glycolysis. And in continue, it turns into fructose 6-phosphate, fructose 1 and 6-bisphosphate, dehydroacetone phosphate, and glyceraldehyde 3-phosphate, 1 and 3-bisphosphate, bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenolpyruvate, and at the end, pyruvate. So this is glycolysis. Now we want to do gluconeogenesis. For the first non-carbohydrate molecule, I want to talk about lactic acid. Lactic acid can enter liver cell and turns into pyruvate. Why? How? When? For example, you're doing intense physical activity and the amount of lactic acid in your amount of lactate and lactic acid in your muscles increase, and you want to get rid out of these lactic acids somehow. What your body do? Your body transport lactic acids from muscles into liver in order to turn them into pyruvate. In a normal state, in a normal situation, we know that pyruvate can enter mitochondria and turn into acetyl coenzyme A. But we're gonna do gluconeogenesis. We don't want to produce ATP from here now. Our main goal and our main purpose in liver is to produce glucose, not to produce ATP. So we're going to turn pyruvate into this 4-carbon molecule here. What is this guy? This guy is oxaloacetate. What's the difference between pyruvate and oxaloacetate? This guy, oxaloacetate, has one additional carbohydrate compared to pyruvate. So let's add a carbon into pyruvate in order to turn it into oxaloacetate, and the enzyme and the regulatory enzyme in this step is pyruvate carboxylase. What this enzyme do? It's going to do carboxylation. It is a carboxylase enzyme. Now here, oxaloacetate in a reversible reaction can turn into malate. In contrast of oxaloacetate, malate can easily move out of the mitochondria. It can easily go out of mitochondria. And in cytosol, in cytoplasm or in cytosol, malate again can turn into oxaloacetate. So if you remember, I said we're gonna do reversible glycolysis. I want to mention again, reversible glycolysis is not gluconeogenesis, and it's not scientifically true, but it helps to remember reactions and the steps and the whole structure and the whole purpose of gluconeogenesis. So oxaloacetate here, oxaloacetate is doing something interesting. Let's take a look here again. We turned lactic acid into pyruvate and turned it into oxaloacetate. Why we didn't turn it to phosphoenolpyruvate in order to these all steps to produce glucose? Because this step, pyruvate, phosphoenolpyruvate to pyruvate, is irreversible reaction. This black arrow is irreversible. These ones are reversible, but this one is irreversible. We cannot turn pyruvate into phosphoenolpyruvate directly. So this is why we help pyruvate to turn into oxaloacetate, malate, and move it out from mitochondria and turn it back to oxaloacetate. Now oxaloacetate can turn into phosphoenolpyruvate. What are these guys' differences? This one has a phosphate group. Okay, this phosphoenolpyruvate has one less carbon compared to oxaloacetate. So let's get rid of a carbon, CO2, and let's add a phosphate group, and we're doing these steps with using the phosphoenolpyruvate carboxykinase. This enzyme, this very important and key regulatory enzyme, helps oxaloacetate to turn into phosphoenolpyruvate. So we have phosphoenolpyruvate now. Phosphoenolpyruvate can easily turn back into 2-phosphoglycerate, 3-phosphoglycerate, 1 and 3-bis-phosphoglycerate, DHAP and glyceraldehyde 3-phosphate, fructose 1 and 6-bisphosphate, and we have another irreversible black arrow here. So what we do here, we have an enzyme called fructose 1 and 6-bisphosphatase. Why we cannot do gluconeogenesis in muscles? Because there are differences in the enzymes in muscles and in livers. For example, we do not have a good enough amount of fructose 1 and 6-bisphosphatase in muscles. So it is nearly impossible to turn fructose 1 and 6-bisphosphate into fructose 6-phosphate in muscles, and this is why we say muscles cannot do gluconeogenesis, because they do not have enough amounts, and in some cases they lack of some enzyme, and in some cases their amount of enzyme is not enough to do gluconeogenesis efficiency. So in liver and kidney, we can use fructose 1 and 6-bisphosphatase to turn fructose 1 and 6-bisphosphate to fructose 6-phosphate. What are the differences between these two molecules? Only this phosphate group. So let's get rid out of this phosphate group. Now we have fructose 6-phosphate. We can easily turn it to glucose 6-phosphate, and another big irreversible arrow, turning glucose 6-phosphate into glucose. So what our body do, our liver cells, our hepatocytes, has, just like many other cells, has a smooth endoplasmic reticulum. Glucose 6-phosphate can enter smooth endoplasmic reticulum, and there is a special, very special enzyme here which can help glucose 6-phosphate to get rid out of this phosphate group, because the only difference between glucose and glucose 6-phosphate is this phosphate group. So this special enzyme in a smooth endoplasmic reticulum helps glucose 6-phosphate to get rid out of this phosphate group, And here we produce glucose, and this glucose using tunnel 1 and tunnel 2, T1 and T2 channels can turn back into cytoplasm, can turn back into cytosol, and eventually can move out of cell. This important and special enzyme is called what? It removes one phosphate group from glucose 6-phosphate. So we call this enzyme glucose 6-phosphatase. Whenever we have phosphatase, it means that we're removing a phosphate group. So this was gluconeogenesis from lactic acid into glucose. But we mentioned that we produce glucose from non-carbohydrate molecules. Do we have any other non-carbohydrate molecule in our cells? Yes, this beautiful big structure right here with one glycerol and 3 fatty acids that we call this 3-acyl glycerol or triglyceride. Triglyceride can do gluconeogenesis for us. How? First and foremost, we should cut these fatty acids from glycerol. So we're doing this with a pathway that we call lipolysis. Lipolysis helps triglycerides to turn into glycerol and free fatty acids. Glycerol can turn into another molecule with one additional phosphate. The difference between these two guys are only this phosphate group. So let's add a phosphate group, and because we're adding a phosphate group, we call this enzyme kinase. What molecule has received phosphate? Glycerol. So we call this enzyme glycerol kinase. Glycerol kinase helps glycerol to turn into glycerol 3-phosphate. And glycerol 3-phosphate can easily turn into dihydroacetate phosphate. And whenever it turns to DHAP, what can it do? It can turn back into F16PP using F16 bisphosphatase to turn into fructose 6-phosphate. It is reversible to glucose 6-phosphate. Glucose 6-phosphate moves into a smooth endoplasmic reticulum and turns into glucose, and glucose can move out easily. So this is how we use lipids and specifically glycerol in order to produce glucose. How about fatty acids? Fatty acids are not very eager to do gluconeogenesis, but they can do a reaction that we totally call beta oxidation, and they can produce acetyl coenzyme A. They can eventually, acetyl CoA eventually can turn into malate. Malate can move out and do all these steps to glucose, but this is not an efficient pathway. So we usually not use free fatty acids in order to do gluconeogenesis. So what other things? We have this beautiful important structure here. This big guy is a protein, and these little circular things here are amino acids. So what can we do? We can do a reaction called proteolysis. Proteolysis breaks proteins. How? It can turn a big structure, a big protein, into amino acids. So whenever we have amino acids can do a reaction with keto acids. For example, an important keto acid in our cells is alpha ketoglutarate. And our example for our amino acid can be alanine. Amino acid and keto acid do a reaction that we call transamination, and they can produce new amino acids like glutamate and modified keto acids. In other words, in this transamination step, keto acid turns into a new amino acid, which in this case we have alpha ketoglutarate, so it turns into glutamate, and our alpha and our amino acid can turn into a modified keto acid. For example, For instance, alanine can turn into modified keto acid, which eventually it can be pyruvate, it can be acetyl coenzyme A, and it can be succinyl CoA, it can be everything. And this amino acid here is the responsible to provide and to produce many substrate of Krebs cycle or even pyruvate. So for example, whenever we use pyruvate from alanine, how it can do gluconeogenesis, pyruvate can again turn into oxaloacetate to malate out of the mitochondria, malate into OAA to PEP using PepCK. It can turn back and do all these steps in order to eventually produce glucose. So this was all about gluconeogenesis. There are some important information you must know. The first and foremost is this, using proteins for gluconeogenesis is not healthy, is not good, because we're using for example, think that we are using our muscles in order to produce glucose. Our proteins are very important structures and very important molecules that has many important steps in our body, and they are not created. Proteins are not created to produce energy, but whenever our body really needs glucose and it cannot use, for example, lactic acid or lipids efficiently in order to produce glucose, in order to keep our blood glucose level in the normal range, unfortunately it uses proteins to turn and to produce glucose. So if you paid attention enough, we have 3 important steps here. These steps, PepCK, fructose 1 and 6-bisphosphatase, and glucose 6-phosphatase, are 3 important key regulatory steps in gluconeogenesis. So remember these 3 enzymes, PepCK, glucose 6-phosphatase, and fructose 1 and 6-bisphosphatase. And they can be regulated mainly by hormones such as glucagon, epinephrine, norepinephrine, cortisol, thyroid hormones, and growth hormone. So this was all about gluconeogenesis. I told you that we're gonna talk about why we're doing gluconeogenesis. I'm gonna answer it right now, because whenever we have low blood glucose level, when we are in hypoglycemia state, when we are in hypoglycemia condition, our body, our liver actually use gluconeogenesis to produce glucose, because glucose is the main energy source of brain, heart, and it is very important to keep it in a normal range. And these reactions, gluconeogenesis, mostly takes place in liver and then in kidney, because, for example, muscles does not have many of these enzymes, fructose 1 and 6-bisphosphatase, PepCK, or glucose 6-phosphatase, especially for glucose 6-phosphatase, in order to produce glucose. Okay guys, I really hope you enjoyed this video. If you have any question, please comment in the comment section. Your like is very important for me. Please hit that thumbs up button and subscribe me if you want to learn about biochemical pathways every day. So see you until next time.